sometimes things 2 or 3 times is not so bad, because you can, as you hear it, you can think about what else you could do. Now, this lecture, you probably have heard nothing about it. It's all about this. Let me put that up. So it's all about this. The first lecture was all about this gene-centric approach. Everything is in the DNA. DNA does nothing. It is a template. Unless you make the RNA of it and you make the protein of it, and the cell has also lipids, and you have proteins that make lipids and put them in the right place, it's all made out away from DNA. DNA is even shed in the nucleus to not interfere with all the things that it actually does. It's like in a bunker, and the rest of the world does what this bunker kind of says, but the rest of the world has to do it. This is the gene-centric view. And there is a guy in another lab here. I asked him, he's a gene-centric person. He works only on DNA and thinks there is nothing else. All the rest comes. Once you have a genome, all the rest is sort of secondary effects of the genome. And if you change the genome, you change these things. There are always 2 points of view from where you look at. The DNA is very important for one thing, and that's heredity. The DNA brings the information to the next generation. So if you're interested in transmission or in heredity or in heritable things and in evolution and in all these things, DNA is, of course, the important thing, because it carries the information from one generation to the other. But we are actually, in developmental biology, we are interested in how cells do things. How do cells make a vascular system? How do cells make a tracheal system? How is the gut built? Why does the gut have different structures? The gut has different structures because the visceral mesoderm gets these structures, it expresses DBP, and then it tells the cells adjacent to it, now you make labile, and labile expressing cells become copper cells, and copper cells have a certain function in the gut. This is the type of logic we would like to understand. It's all proteins. And that's, so as much as the first lecture was gene-centric, which is of course wrong or partly wrong, as much the second lecture is protein-centric. So I'll try to stress the importance of proteins at the expense of DNA a bit. So it's one side of the coin and it's the other side of the coin, and a coin always has 2 sides. You can't have a single-sided coin, really. So it will be protein-centric. It's also a lecture that I adopted and changed a bit, but this was in the context of flies. And since the block course is about flies, and you will not hear from others about flies, I will make it, of course, protein-centric and fly-centric as well. So genetic research in flies has a long history, in C. elegans as well, but it has a long history. Started with Morgan, as Martin said, in 1905. The white mutation was the first one identified. People realized that information is on chromosomes using flies. People realized that chromosomes can be changed, and DNA can be changed by nerve gas, by X-rays, by radiation. So Ed Lewis, the guy who won the Nobel Prize, he brought himself to the first detonation of the atomic bombs in the US. They brought flies there, put them beside the explosion sites, and collected them again later and asked, do they have now changes in their genome? And that turned out to be the case. So it was clear that radioactivity would alter the genome. So lots of things were done with flies. Easy to keep, very important. Money is always an issue, money and space. Large progeny, important to do genetics. If you only have 2 progeny and one has red and the other one, 2 have red eyes, you cannot do the statistics. Large progeny is very important. Few chromosome helps. Nowadays, sometimes I think we would be happy to have more chromosomes, because in a fly where we want to ask, is DBP receptor important? We need 2 thick vein alleles, for example, or 2 punt alleles. Then we want to have something driving GAL4 in a certain tissue. Then we want to have UAS, to which GAL4 binds, driving a certain protein. So that makes 2 other chromosomes where we have to have a P element. Then we want to have a reporter, and so on. So as more as we go, maybe few chromosomes will turn out to be a disadvantage. But of course, we can recombine things, but that takes one more generation. We load the genome now of lice usually with several transgenic constructs at the same time, so that we can control gene expression in time and space and have a readout, GFP, and so on. And then a control readout in red. So a few chromosomes, yes, sometimes it's a disadvantage nowadays. Extensive genetic toolkit with P elements that we can use, and then with this GAL4 system, that was a synthetic transcriptional activator response system. And this toolbox is changed every day, every year. There are many more things in the toolbox. Now it's kind of dramatic because we cannot order flies from the US, from the stock center. They haven't made it, the last ones we ordered, so we are kind of down. It's not so important. Some more important things about this, about coronavirus. Large scientific community, it's also very important. Thomas Hunt Morgan got a Nobel Prize, many Nobel Prizes for flies. It's all for genetics. That's the point I want to make. Thomas Hunt Morgan, discovery concerning the role played by chromosomes in heredity. Muller, discovery of the production of mutations by means of X-rays, got the Nobel Prize. Janni Nüsslein, Wisch Aus, these two people who were here in the room where I am now, studying with Walter Gehring, they got the Nobel Prize for the genetic control of early embryonic development. Genetic control, you see, it's all about genetics. Then Jules Hoffmann from Strasbourg won the Nobel Prize for discoveries concerning the activation of the innate immunity. So immunity. Cells invertebrates don't have B cells, but they have an innate immunity identified first in flies, and then the same molecules, same genes were also found to be associated with this in mammals and humans. And then the last Nobel Prize, 2017, was to Hall, Rosbach, and Young for the circadian rhythm. All of these were genetic studies that led to the identification of important mechanisms that later on were found to be conserved in other organisms. So genetics were really important. For over 100 years, the manipulate the gene has been used to alter the encoded protein. If we want to have a thick vein receptor that is activated, we can now in the genome change one amino acid, D, Q in thick vein to D. This is thick vein QD. And then we have an activated version of the receptor that signals independent of ligand. Using CRISPR, we can introduce this mutation now into flies. This would, of course, be lethal because all cells have an activated thick vein receptor from the first cell to any progeny of this cell, and it would be lethal. So, yes, we can modify the genome and modify proteins, but since the genome is conservatively replicated in each cell has these modifications. So, to use the genome to work with proteins is necessary, usually for lots of function, but it's not very easy to really manipulate proteins. But of course, we didn't know anything else. We don't know how to modify proteins. So let's do it with a genome, and we can do a lot of things with CRISPR. But actually, we can't acutely modify proteins. But yeah, ask many developmental biologists, they don't care because they have enough work to do with what they have to do now. So for many of them, maybe no limitations. As I said, CRISPR will maybe not be made better. So here we are. Let's use what we have now and we didn't have before, and then we can do avant-garde science because this science could not have been done 5 years ago. But still, we don't really do what we want to do. So I did my postdoc with Walter Gehring, this famous developmental biologist. Martin Müller, he did his doctoral studies with Gehring in 1988 or so. And Gehring is a developmental biologist. He was interested in eye formation. He died, unfortunately. This is a few weeks before he died. He gave a big lecture here in in Basel. He was a real zoologist, developmental biologist. How do you make organs and all that stuff? But he was also mostly into genetics and not really into internal organs. He was into homeotic genes. And what I wanted to do is to study how organs form. This is the tracheal system. How does the lung form, for example? How does how do blood vessels form in zebrafish? We'll hear more about this later. Tracheal development you don't hear all that much about, because you will hear about vessel development tomorrow. But this was sort of how are three-dimensional structures generated from the linear DNA molecule. That was what I always wanted to kind of understand, or try to contribute to understanding. Very little is known about the lung, for example, because flies don't have a lung, and zebrafish doesn't have a lung. So you have to do it with mouse, and mouse embryos you cannot really work with all that much because you kill the mother. So a lot of organs, there is not that much known of how they develop. And what we wanted to do, or what developmental biologists who are not interested necessarily only in the genetics, but in the cell biology, and again, the cell is the basic unit. This is the cell does the job. The nucleus helps it to do a good job and tells it which proteins are in the cytoplasm and which proteins are allowed to do the job. But then the proteins have to sort it out themselves. The DNA doesn't help them to do the job. So the cell functions. If you remove the nucleus, the cell still functions very well. Eventually, it will not be able to maintain its protein production because the nucleus also makes the RNA. But the job is done by the cytoplasm. You can enucleate cells. They will still migrate. They will still do what they actually should do, but they will run out of fuel at some point. So what we wanted to do, and other developmental biologists too, of course, was to use flies or fish and to understand cells in vivo. What do cells do? What do cells do to generate a certain organ? We all start with one cell. And maybe the primordia of this organ, the primordia of the wing imaginal disc, once it's set aside, is about 30 cells in the embryo. And at the end, you have a wing and the thorax and all the functional structures. What happens in between? Why does the wing have this shape? It's all about cells. So we need to do in vivo cell biology. And the question we said from the very beginning, it was only possible when we started this to do it this way, is how do individual cells behave? There was no GFP, we couldn't even film cells. So that was, of course, very difficult then. But when GFP came up, when I started my own lab, GFP was there, so we could use it. And then we could start to film cells, document what cells do, and sort of see, yes, one cell, two, four, one moves away, one does this and that. At the end, we have an organ. How do individual cells behave? How do signaling systems control this behavior? What are the effectors of signaling systems? Cytoplasmic effectors, we didn't talk about at all, but the cytoplasmic effectors make a cell move, divide, and so on. What are the forces? This is one of the most interesting things. If you look at the movie, you see things happening, but without force, nothing would happen. If you kill force, then it's a frozen picture. Nothing happens. So forces are very important, but you need forces to do cell division. So if you take the forces away, if you take the actomyosin away, the force generated, then you can look at the movie and nothing happens. So without force, architecture will not change, cells will not divide, nothing happens. But how do you use genetics to remove forces? If you inactivate actomyosin, the cells don't divide, you don't get a clone, you don't get an animal, you don't get anything. So one of the major drivers of generating three-dimensional shapes, structures, and animals is forces, and genetically we can do very little or close to nothing to analyze forces. So genome editing has a lot of limitations. And that's what I want to talk about now. So this, how can I do this? I cannot do that. Why does that not work? It worked yesterday. Let me see whether this is I'm sorry.
Marcus, maybe you should try opening just the original file, because maybe it was inserted, not in.
Yeah, let me briefly have a look at it. It's not, also, it would drive 1 point home. It's not so important. 22, you see, I did upload the modified version. This is really a bit of pity. Okay, look what we do. Give me 5, I'm sorry, but it's a bit of pity to it's more difficult for me to drive home my point if I don't have these movies. There are not so many, but give me 3 minutes. We make let's make a 5-minute break, and I'll try to organize it. Is that okay? Thumbs up. Seems to be okay for some people. Thank you. So I'm sorry, give me 5 minutes. So we will be back at, now it's 5 minutes is 44. And then hopefully this will work.